Hi, everyone. My name is Azka. I recently defended, so I'll be starting as a postdoc at Frost Lab at Dartmouth College. And I'll be speaking about cytokine activity estimation for human and mouse models using SCAPE. So a key motivation for cytokine activity estimation is that it's a pressing problem. So cytokines are implicated in a number of different conditions, including autoimmune disorders like rheumatoid arthritis and also COVID-19. And they're also one of the central agents in immune cell signaling. Second, existing cell-to-cell interaction methods are largely limited because they, one, primarily function at the cell type, population, or cluster level. And second, a lot of methods don't distinguish between interaction potential, where the receptor is present, and interaction activity, where the receptor is present and bound by the ligand, based on expression from molecules that are either downstream of the receptor or on the molecular pathway of the receptor. And thirdly, current methods like NicheNet, for example, usually result in a ranked list of ligand activity as opposed to single cell resolution activity predictions. So one of the key innovations for our method was that we wanted to provide single cell level estimates of cytokine activity estimation. So in brief, I'll start out by providing a schematic of the SCAPE method followed by some initial results, and then discuss an implementation of the SCAPE method to mouse models followed by some of the initial results for that. And lastly, venturing in future directions. So the first step for the SCAPE method is gene sets construction using the cytosig-based gene sets. The second key step is gene set scoring via modified variance-adjusted Mahalanobis method, VAM. And third, we are providing an extensive evaluation on both public transcriptomics data and also on stimulated data. So for the method schematic, the first step is gene set construction. And here we are utilizing the Cytosig database, which is published in Nature Methods. And for the Cytosig database, the authors utilize 4 different sources of bulk transcriptomics data sets, which are indicated up here on top. So GEO is one of them. And then they construct these expression matrices for each one of the 43 cytokines that they have currently documented. And they indicate the genes that are both positively and negatively differentially expressed. So in the end, we have a database that we can then reference for subsequent gene set scoring mechanisms, where for each one of the 41 or 43 cytokines, we have a list of positively and negatively differentially expressed genes along with the weights. So this is for a human equivalent, and there is a mouse equivalent that I'll discuss as a database that I think really takes some of these analyses up a notch based on real transcriptomic single cell signatures. But here, this is for human bulk. The second step is gene set scoring. Here we leverage and modify a variance-adjusted Mahalanobis technique, which is currently only applicable for positive weighted scores. So we are now adjusting for cell-specific negative weighted scores, which can potentially account for suppression in the cytokine activity. So as the input to the matrices, we take normalized single-cell RNA sequencing matrix, we take the gene sets weighted membership matrix, which comes from the previous step, and then we construct a modified gamma CDF scores for the positive and negative weighted gene sets and average those. And this is utilizing, again, a modification of the VAM method, which was recently published. And you can read more about it in our streak method and also in our VAM procedures. And so two of the key ways in which this modification differs from the canonical Mahalanobis distance is that one, it computes distances from the mean. And second, it only discounts the technical variation. The biological covariation is preserved, which I think is one of the key steps for providing more biologically interpretable signatures that can then be utilized for different tasks like cytokine activity estimation. And for the third step, we performed evaluation on both public transcriptomics data and also on stimulated signature data, which I think is key because currently we don't have single cell level transcriptomic signatures for humans. So we need to generate stimulation signatures that tell us what the transcriptomic signature looks like at the single cell data when we have exposure for each one of our 41 or 43 cytokines. So here we are looking at 41 cytokines. And then we are simulating single cell RNA sequencing data using the correlation structure of a resting cell single cell RNA sequencing data, which is a Knogemes et al. Data set. You can read more further about it. And then I have some pseudocode here leveraging an idea of using copulas as a framework to generate simulated correlated data. And here I briefly go into, we are coming up with the updated mu's by multiplying the prior mu gene by the log fold change associated with the Cytosig database. We are setting the overdispersion parameter to 100, generating multivariate random normal data, generating random uniform, and then using the quantile function to generate marginal negative binomial data. And of course, we can consider variations of it by considering different types of distributed single cell data, but this is for simplicity, is what we have currently. In terms of our results for the simulated data, so this is the SCAPE method using the cytosig trained gene sets. We get this initial matrix where the rows represent the simulation model and columns represent the cytokine whose activity is being estimated by SCAPE. So in the case where the ordering of the rows and the columns is identical, we should expect to see a diagonal structure, which indicates that the cytokine that's used to simulate the data is the cytokine with the largest activity, which intuitively makes sense, right? So the cells that are being stimulated should have a higher activity for that cytokine. And that is what we expect when using the SCAPE method. And this is, again, simulated single-cell RNA sequencing data. So this is what we expect for the SCAPE method using the cytosig trained gene sets. And next, so we are comparing our SCAPE method against the cytosig implementation, which is cytosig's own cytokine activity implementation using a linear regression model. It's a ridge regression-based technique. Against niche-net model, a SCAPE method using the reactome trained gene sets instead, which are unweighted, and also a variation of the RNA normalization approach, which looks at the receptor equivalent of each cytokine. So again, what we observe is about 41 cytokines, which are 41 is what we had studied, show a presence of a diagonal structure. TNF-alpha has somewhat of a lighter structure, but we can further look into why that might be the case, but it's still higher than the score for the comparative methods. In comparison, about 30 out of 41 cytokines show a diagonal structure for the cytosig trained gene sets and for the cytosig cytokine activity estimation framework. And so we have our preprint on bioRxiv, so you can look at the corresponding matrices for the rest of the estimation methods there as well. Next, we validated using rail perturbation data sets. So we have 3 different data sets, and for 2 of the data sets, the IL-4 and IL-13, we have stimulation signatures, and then for IL-10, we have a blockade condition data set. So for IL-4, for example, we should expect to see a higher signature for the IL-4 stimulated activity, and that is what we expect. So IL-4 has about an average predicted cytokine activity of about 0.75, which is higher than the rest of the cytokines. And again, in the main manuscript, you can see that we compare against all 41 cytokines. Here, I'm just showing a subset of those. Similarly, for IL-13, we see a very high signature for the chronic IL-13 data. And in comparison for IL-10, which is a blockade condition, we see a lower signal or lower average predicted activity in comparison to the stimulated category. In comparison for the cytosig's activity prediction framework, we don't generally see a lot of expression heterogeneity for IL-4, for example, and even for IL-13 and also for IL-10, where we are not generally seeing a correct either upregulated pattern on capturing of those patterns via the average predicted cytokine activity or the downregulated pattern as in the case with IL-10. Next, I'll briefly go into our SCAPE for mouse schematic. So this was a data set that was recently published in Nature by researchers from the Broad Institute who administered about 86 different cytokines in mouse with three mice per each cytokine replicate. And then they collected and drained lymph nodes and sequenced single-cell RNA sequencing data set. So this data set is key because now we have single-cell level transcriptomic signatures for cytokine-stimulated data, which we can then leverage for predictive modeling. And then I think it's also important because we can consider taking these models and administering them in clinical settings where for a given patient transcriptomic signature data, we should be able to see what are the cytokines that are most likely responsible for the given signature. So we first essentially demultiplex the data because there are about 45 different data sets and multiple cytokines for each data. So we demultiplexed it, pooled it together. There are about a little over 350,000 cells. And then we are performing differential expression analysis to get a list of genes that are both positively and negatively differentially expressed, and then using those genes in our gene set scoring technique, which I had described earlier, which is a modified version of the VAM approach. And then we are validating through a stratified 5-fold cross-validation approach, and also generating ROC curves and AUC scores to evaluate the predictive accuracy of our models. We are comparing against cytokine activity estimation techniques, which include the IREA. So this is the tool that's created by the authors themselves. It's not currently functional as an R package. So we have considered an implementation of it based on the author's description. And then we are also comparing against the SARAT and UCell gene set scoring functionalities and also cytokine activity prediction framework for our SCAPE for mouse model, which can take 4 different forms depending on what we are either if we are training our model against all cytokines or against just the PBS unstimulated control. And this applies to both differential expression analysis and also evaluation. So this first model and the results that I'm showing, this is for the model that's trained against PBS and evaluated on PBS. So I have 4 different evaluative methods that I'm comparing SCAPE against. One is UCell, Seurat, IREA, and then SCAPE. So the higher the AUC, the more greenish and yellowish hue we should expect, and the lower the AUC, conversely, the more reddish and orange tones we should expect. So overall, we can quantify, we can see that SCAPE works well in practice, followed by the IREA strategy. And then we can quantify the individual method value for each one of these techniques by quantifying the proportion of cytokines that are elevated or that show a higher AUC for each method compared to the rest of the estimation strategies. So we can see, for example, for the model that's trained against PBS and evaluated against PBS, there are about 69 cytokines that show a higher average activity or that have a higher AUC when evaluated using the SCAPE method as compared to the rest of the estimation strategies. In terms of future directions and limitations, so one, we can consider hierarchically determining groups of differentially expressed genes, or also considering more recently proposed techniques for differential expression analysis that can quantify differences between the control or the unstimulated and the stimulated data sets. And then we can second quantify a list of cytokines that are transcriptionally most similar to the cytokine of interest based on the common set of differentially expressed genes. So these first 2 future directions are addressing a key point, which is that a lot of these cytokines have pleiotropic and redundant functionality. So we want to be able to model the simultaneous effect of each one of the cytokines, accounting for and including another cytokine. Third, I think it would be very valuable to extend this technique to perform multi-class classification via neural network models. And fourth, we would want to extend an implementation of the SCAPE for mouse method to other data sets, as well as also including cell type adjusted gene sets. And fifth, I think we are currently only incorporating expression from about 30, both positively and negatively differentially expressed genes. So we would like to extend this framework to accounting for more than that genes. I think that would help improve the performance and the AUC scores further. In terms of the acknowledgements, I would like to thank the Frost Lab alumni, my funding, as well as also the Quantitative Biomedical Sciences program at Dartmouth College, and collaborators and correspondents with the Cytosig team and the IREA team. Thank you, and I'm happy to take any questions now.
So you mentioned that you partitioned the covariance into biological and technical. Could you go into detail about how you do that?
Yeah, that's a good question. So we personally didn't do that for the simulation data simulation strategy, but we did do that for the variance adjusted Mahalanobis method. Is that what your question?
Yeah.
Yeah, so that uses the variance decomposition function from Seurat. I can go into the theoretical details of that, but I think I'm not really familiar a lot with in terms of how it's exactly implemented. But we considered a variation from Seurat that helped decompose the variation and the technical and the biological variation. But that's a good question. Thank you.
Any other questions? So conceptually, what is the key difference between the Cytosig's own tool and SCAPE?
Yeah, that's a good question. So Cytosig's own tool also looks at the composite effects of multiple cytokines in their linear regression model. So I think that's one of the key reasons why it potentially doesn't perform as well in comparison to our current model, which is looking at each cytokine individually. But I think their method conversely does help address the limitation that a lot of these cytokines have pleiotropic and redundant functionality. So I think maybe there's more interpretive benefit to their method, whereas I think our method has more predictive performance and accuracy. In terms of that, it might have more benefit.